Hi, I'm Dan Koster, a PhD candidate in Ron Shamir's group and an MD student. And today I'll show you a method we developed for the detection of cancer risk based on the electronic medical records, focusing on our results for breast cancer. So breast cancer is the most common cancer among women. Therefore, different screening approaches have been developed, like clinical breast exam and mammography. When resulted in an abnormal finding, the patient would require to conduct a follow-up test, such as ultrasound, MRI, or biopsy. That raises an issue, since those tests are expensive, labor-intensive, might be inconvenient, and their accuracy is limited. Therefore, we suggest an alternative approach by utilizing electronic medical records of healthy women to predict their cancer risk. In order to achieve this goal, we work on a quite unique dataset of Tel Aviv Medical Center that contains routine checkups of individuals that were occurred as part of their insurance coverage. Each patient is supposed to visit the medical center every 2 to 3 years. And during each visit, more than 600 features are collected, such as different lab tests, sports, and nutrition habits. Our goal consists of 11,000 checkups of more than 6,000 healthy women. Using the Israeli Cancer Registry, we detected 77 women which developed breast cancer less than 2 years since their last visit. Our method uses survival analysis, but instead of death, we define the failure event as a cancer diagnosis. So for example, we looked at subject A. Since her first visit to the medical center until its cancer diagnosis date. And this is her failure event. Or subject B, which is also being followed since her first visit, but by the end of our study, she's cancer-free, and hence right sensor. Since in our code we have multiple visits per patient, we use pseudo-objective transformation to handle it. So now we can define our survival curve, ST, as the probability that the patient is cancer-free up to time t. Given the survival function of our dataset, we can now construct a decision tree with nodes that contain survival functions. For example, splitting the samples in a way that all the samples in which the white blood cells value is lower than 7 will be on the left node, and all the rest will be on the right node. Each of these nodes induces its survival function. Then we will use the log rank score test to check the significance of their difference. So in that manner, we iterate over all the features and their cutoffs to select the pair which yields the lowest p-value. We will keep building the tree, and our stopping criteria would be when there is no statistically significant split. Our approach is to use ensemble of survival trees. By an ensemble, I mean that instead of one tree, we generate 500 trees. But each node of the tree, instead of iterate over all the available features and examine only their cutoffs in order to split the node, after building those trees, a new sample, we randomly select a small number of features to examine only their cutoffs in order to split the node, like in random forest. After building those trees, a new sample will traverse all of them, and finally arrive to 500 leaf nodes that contains the survival function. Then we average them to one survival function per sample. Using the predicted survival curve, we can now predict the probability that the patient will remain cancer-free at different time intervals from their last visit. For example, here, in the next year, and we define the risk score as 1 minus this probability. The x-axis represents the different time interval in which we evaluated our method. The y-axis is the average AUROC. Our method, in blue, was compared to Cox regression, a random forest classifier, a single survival tree, and to the performance of Gale models that were reported in the literature. As we can see, our results outperform the others' methods until a time interval of 1.5 years. We also evaluated our model in comparison to common screening tests. We looked at all the women that were diagnosed with cancer and during their visit conducted the CBE or mammography. With clinicians, we created a code that automatically classifies each textual summary of the test into normal or abnormal finding. This is the number of days between their last visit until the cancer diagnosis. That's the risk score that was given by our model, and their finding in the screening test. We can see that the women with the highest risk score were not detected by other screen tests. Thank you very much, and have a great conference.